So now we get to the heart of the matter. Pun fully intended, the circulatory system. Heart pumps blood all over the body, and there's a lot of good chemistry in blood, from osmosis to just how water works. Why is our body mostly water? So we'll talk about that. We'll talk about a little bit of inheritance. But first, let's just talk about the physiology of the circulatory system itself. So why is the heart so important? Why, if your heart stops beating, does death follow really soon? Because the heart distributes blood, and blood has all the good stuff in it. Carries wastes away from the cells too, of course. But think about it. Your heart pumps blood down to your digestive system. There it picks up lots of nutrients and brings it back to the heart. And once in the heart, it distributes it all around the body. Brain, muscles, everywhere. There's actually a separate circuit for the lungs. So the blood coming back from those cells goes first to the lungs. You breathe in, oxygen goes into that blood, carbon dioxide comes out, and then goes back to the heart to go around the whole rest of the body. And for our purposes, that explains a whole lot. It's got nutrients, it's got oxygen, now it gets to the cells and helps them out. Depending on what we're doing, the circulatory system can adjust because our arteries have muscles around them, and they can constrict and cut off blood flow, or at least decrease it. Like when you're resting, you don't need a lot of blood going to the muscles. And they can dilate and deliver more blood to different areas. So depending on what you're doing, it's pretty adjustable, delivers more or less energy and nutrients to the cells as they need it. Well, first let's get into some of the chemistry, talking about water, hydrogen bonding, and also pH. Well, here we return to this idea that most molecules in our body are composed of polar covalent bonds, and that separates them from those that have mostly nonpolar bonds like the lipids. Water, being the most abundant molecule in the body, remember, has this slightly negative charge on the oxygen and slightly positive charges on the hydrogens. And so that makes it kind of lopsided, which is what we summarize as polar. Well, that's true of other water molecules as well. And so if I were to draw that one in, then the attraction between the partially positive hydrogen ion and partially negative oxygen creates this attraction. I won't call it a bond, but I'll say that it is hydrogen bonding. You will sometimes hear it referred to as a hydrogen bond. I just don't want to confuse the issue. These electron sharings are bonds, so are the attraction between the positive and negative charges of an ionic bond. But the hydrogen bonding is more of a temporary attraction between different molecules. And so there's also this feature where, since it's partially positive charges, there's an attraction with that negative chloride ion. And even if you had something like a glucose molecule, remember each of those has an oxygen and a hydrogen, that hydrogen is going to have a partially positive charge and be attracted to that oxygen as well. So hydrogen bonding, anytime you have an oxygen or a nitrogen with a hydrogen, you're going to have that polar bond set up to make hydrogen bonding possible. And that holds so many different things together. Remember, it holds the two strands of DNA together. It holds the water molecules together so they don't just float apart. And it means that polar and ionic compounds can dissolve in water really easily because of all this hydrogen bonding and polar bonds. All right, let's return briefly to pH just for a moment to talk about buffers a little more. We've done a lab on this, or we'll do that soon. So what is a buffer? Remember, it's a mixture, so it's a mix of a weak acid and a weak base. So if you put both of those together, you have a buffer. If this solution starts to become more acidic, that means there's more hydrogen ions, right? Well, the buffer mixture can absorb those hydrogen ions. So they just get absorbed into the mixture, mostly by this weak base component, because bases can absorb hydrogen ions, and that pushes the pH back toward neutral. If we do the opposite and we start adding hydroxide ions and making the solution more basic, then the weak acid part of the buffer mixture can give up a hydrogen ion, which would make it more acidic and push it back toward neutral. So that's how they work. It's actually fairly basic, and that has a lot to do with the chemical reactions in solution, namely that they are at equilibrium. So to really generalize this, I've made these components A, B, and C, just showing that we can combine them in different ways. Remember, chemical reactions are just breaking and reforming bonds. And that reaction can occur so that A and B get broken up and attach A and C together. That's not the important part here. What I want to focus on is the fact that they can be broken back up and A and B can get back together. And so we show this as a reversible reaction with those equation arrows pointing in both directions. So what that means is that this weak acid, weak base buffer mixture is an example of this, right? That if the solution becomes more acidic, then the reaction gets pushed one direction, and if it becomes more basic, it can be pushed the other direction to help stabilize the pH. Following up with water, it's important when we talk about water that we understand how it moves around the body. So obviously in the circulatory system, that heart pumping is one way of moving water long distances really fast. But on a cellular level, once we shrink way down, then what drives water movement is the solutes, the salts and other things dissolved, that water then spreads out to dissolve everything evenly, and that process is osmosis. So let's look at the cell level and talk about osmosis. Let's finish up with the cells and go to the bottom here. We've talked about lots of different membrane transport methods already. And so we need to add one final one to our simple methods of membrane transport and recognize that inside of the cell, there's lots of water. Outside of the cell, there's lots of water, and that water can move in and out of the cell. When water moves in and out of this through the cell membrane, that is osmosis. Osmosis works in some ways kind of the same as all the other types. It wants to move from a high concentration to a low concentration, but since water is so abundant, this works a little differently. So what would make water a low concentration? Well, what if we had lots of ions inside the cell and we had fewer ions outside the cell? If that was the case, then the water concentration inside the cell is lower because there's more other stuff. And so the water is going to want to move in following the solutes. Remember that phrase, water follows salt, and you're in pretty good shape with that. We can skip all the way to the end to talk about physiology. Physiology of the circulatory system. We center a lot of this around blood, and blood is a major vehicle. It carries materials to and away from cells. So we've emphasized that it has lots of water in that. And that water has an interesting property in that the water itself carries a lot of heat energy. It stores that energy in its bonds, and it can release it as heat. So where the blood flows, more heat goes. When your face turns red, there's more blood there, and you feel warm. But dissolved in that water are lots of different things. We have glucose, as you well know, you got blood glucose levels that some people need to monitor. You have a variety of proteins being delivered to the cells. And then there's a variety of ions, including sodium ions. That's probably a big one. There are cells, both red blood cells and white blood cells. And then, of course, there's oxygen and carbon dioxide that vary a bit in concentration. The arteries going to the cells have lots of oxygen, which makes the blood more red. And there's not much in the veins. There's more CO2, which makes it appear a little more bluish. So there's the components of blood. Let's go back to the heart and a little bit about flow down gradients. When that heart muscle contracts, it squeezes in on this chamber and creates high pressure, kind of like the diaphragm up here did before. And so that causes the blood to exit the heart. And out here in the vein or the arteries, there's lower pressure. Let's call that medium. And then let's keep following that down the line. As we get down toward the cells, it's fairly low pressure down here, and so blood keeps flowing that direction. And then as it passes the cells, the arteries turn into capillaries, which then gather back up and form veins. And the veins have very low pressure, and so that very low pressure causes it to return to the heart. And so the blood flows back in here, and this is a fairly relaxed chamber that's also very low in pressure. And so that happens, then it closes off and squeezes and creates these pressure gradients. So the muscles create pressure gradients that move the blood around. Simple enough. Okay, so we get a little bit into inheritance here as well. Not on the concept map, but worth talking about briefly. So it works the same as all other types of inheritance in terms of the Punnett square, but this one's a little strange in terms of codominance. It's actually both codominant and simple dominance. Simple dominance is the one we took on before, where one allele is dominant over another one. But we even have something else going on. Instead of just two alleles, a dominant and a recessive, we actually have three in this case. So we have A and B, they're both dominant, and they can be dominant at the same time. That's where the codominance comes in. Both of them are dominant to O. So O is the recessive allele. So we have a weird way of noting this. It's not capitals and lowercase. We just choose three different letters. So what these things are is little markers, these protein carbohydrate mixes on the surface of your blood cells, but saying, hey, basically, this is me, immune system. These are my cells. These are my markers. And so on the surface of this blood cell, some little markers are type A and other markers are type B. And if you have both of them on the same cell, then you're type AB blood. If you only had the A's, then you'd be type A. Obviously, if you only had the B's, you'd be type B. If you had none, then you'd be type O. All right, so those are the alleles. How does inheritance work? Let's take a look. Well, the same setup as before. Really familiar Punnett square. Letters look a little bit different. This would be a cross between someone with type AB blood and type B blood. So the phenotypes, or the expression, is just type something. Type AB, type O, type A. So if you have an A and a B allele, then you're type AB. Remember, you got two alleles, you can only pass on one. So it's either going to be A or B. If you're type B blood, well, obviously you could have two B alleles, but since the O we kind of think of as a blank, it's essentially recessive, then you appear as just type B. So this person had a genotype of BO, but a phenotype of type B blood. Filling in the Punnett square works the same as it has in the past. Then we have a pretty good mix of genotypes in the offspring. And again, we just look back to our legend over here to figure out what blood type we would call them. Obviously, this one would be type AB blood. Type B, easy enough. This one is also type B, because that O is recessive. And then this one would be type A. So they have three different phenotypes of their offspring. So same sort of system, but the way you interpret it is a little different because that codominance thing going on.